I'll be very scared if I like trailed up in a meeting and I opened the XKCD comic.
Is there anything I can do about that?
No, you're fine. I was just.
Oh, great. Oh.
I legitimately thought that there was a tiny TV in there.
Yeah, there is that.
It's very tiny reflective TV. Oh my God, I can see myself. Welcome everyone. We are already streaming on YouTube, so just so you know, if you do not want to be on YouTube with your camera on, this is a good time to turn it off. And just waiting for folks to come in. Oh, lots of people coming in. Sorry, I can't catch everyone's name that fast. But hi everyone, good morning, afternoon, evening, depending on where you are. I believe we also have the Otter active already. Is that true, Malvika? Perfect, thank you. And the link is going to the, I hope I got the right link in the chat. Yeah, perfect. Thank you, Yo. We are just 1 minute away from starting, but we have, if you are a guest or a colleague of a friend of OLS, very, very warm welcome for joining us today. Please do put your name, project, social handles on our Google Doc down on page 2, if you would like to. And there's also an icebreaker question asking you to share your favorite pastime when you were 10, and is it still something that you still do? I love George's making up weird languages with your sister. I definitely do that too. Did that sometime. Animal symbols though. I'm just gonna put the Google Doc again. Okay, we are at 30 past the hour, 31 now. I guess I'll make a start. All right, welcome to, I think, the final graduation call of OLS. Mixed feelings here, but welcome, welcome, welcome everyone who joined us before, who are new to OLS. Just so you know, we're already streaming on YouTube. So if you do not want to have your face on the YouTube video, this is a good time to turn that off. But this is done so that, you know, other folks who would like to participate can reach a wider audience as well with the fantastic presentations that we're going to hear today from our awesome OLS participants. So today we're going to have 10 presentations, I believe. So we're going to try and be quite strict with time, but before we head into the presentations, as with any other call, we'd like to remind you that the code of conduct is active. For this call, we ask you to be respectful and inclusive. And if you have any questions regarding that, if you witness any behaviors that violates that, please do contact the OLS organizing team, Yo, Malvika, Berenice. Okay, did I miss something? Just to check. Missed an introduction. For those of you who are just joining us, my name is Emmy. I'm a co-host in OLS 2, and we're joined today also by, of course, our wonderful participants, but above all, our organizers, Yo, Malvika, who've been the creator of this thing, and also Berenice. Hi, you're watching when you're watching this recording. Lots of love. Let's start with our first presentation for today. Stephen, whenever you're ready.
All right. Okay, great. Let me get this up. I got it on Google Slides. Click to share screen. All right. Okay, so There it is right over here. Okay, great. Loading. And caption. All right. So good morning, everyone, or afternoon. So what I want to talk a little bit about is my project with the new synthetic biology chassis for open domain use. So what is synthetic biology? It's really the reprogramming of any cells of interest that you're working with, most typically bacteria, yeast, or algae, to perform new functions that you don't find in them naturally, i.e. in the wild. So I wanted to give a couple of examples. Yeast, which is the microorganism that we use in baking products, alcohol, they typically just make carbon dioxide and so forth. But what happens here, you can actually engineer yeast to produce vitamin A. So if you can use the yeast to produce vitamin A in a cupcake, then that cupcake is actually healthier for you, right? Bacteria, you can also use them to sense environmental pollution, and you can also design functions in them to help detoxify these pollution from the environment. Even in cases where algae, unicellular algae, they're using that as a platform to create therapeutic proteins like antibodies against specific viruses and pathogens. So synthetic biology is really about making these cells do new and interesting things, most often for the quality of human health. So how does this work? So in this case, I'm just using the bacterial version here, but I want to make kind of an analogy to a smartphone, which most people have. So with your smartphone, you have the exterior case that holds all the physical parts and the information inside the smartphone. And likewise, in the bacteria, you have a cell membrane and cell wall that contains all the parts and information inside. In synthetic biology, we call that a chassis. Now, for your smartphone, you have an operating system that is able to let the smartphone run and turn on and so forth. That's analogous to bacterial DNA. So bacterial DNA has all the information to make all the parts run the bacteria. But what if you want your smartphone and your bacteria to do new things? Well, your smartphone, what you do, you download apps, install the apps, and your smartphone can do new function. Similarly, with bacteria, you have plasmids, which are these round parts right here. So it contains additional information to make new parts, and the bacteria can do new things. So synthetic biology actually uses a lot of plasmids, manipulation of this to generate new function for cell in synthetic biology. So with this, I wanted to really talk about why I came up with the design of an open domain synthetic biology chassis. So inspiration was the Free Gene project at Stanford. They have open source plasmid for synthetic biology application for a bacteria known as E. coli and also for yeast. And people are working on other types of organisms as well. For me, what I want to do, I want to use this to build an open domain plasmid for a bacteria known as Bacillus cetulis. It has been used a little bit in synthetic biology, so that itself is not new. But what is new is that with the Free Gene project, they're using a lot of other different types of modular reagents and so forth to build those pathways in those microorganisms. Whereas with my plasmid, I am focusing on public domain biotechnology, which is older technologies where the patents have expired. It's been published as public domain reagents through Dr. Jenny Malloy, Open Bioeconomy. So at the moment, it's just a collection of reagents that anyone can get. But this vector, this chassis will be the first platform to actually use those reagents with that in the interest of making things as free and accessible as possible. So with that in mind, the future goals, I just want to mention that once we produce the plasmid, make sure it works in bacteria, there will be further iterations of improvement and expansion of the functionality of the plasmid. And the other goal that I want to have here is we need to be able to streamline synthetic biology. And by that, we want to make synthetic biology accessible, not only for scientists, but non-scientists as well. And in order to build a open bioeconomy community with not only scientists, but the rest of the people in the world, we need to lower the learning curve of how to make these synthetic bacteria or yeast and so forth, because a lot of people are still intimidated by the things they have to learn how to do before they can be successful. And that is very integral or very crucial for increasing the autonomy and sovereignty of individuals and communities around the world, particularly in developing countries where they don't have the financial resources to buy these materials or the stability of keeping biological reagents in low temperatures like minus 80 degrees Celsius. So with that in mind, I just want to thank you for watching. Please feel free to connect with me at Twitter, email, or OLL Slack. And thank you for everyone that's helped me in this journey.
Thank you very much, Stephen. Round of virtual applause for Stephen.
All right. Okay.
Thank you so much.
There we go. Thank you.
Yeah, lots of folks love the analogy you made between the smartphones and the adaptation of bacteria. And if you have any further comments or a question for Stephen, please do put them on the Google Doc. Sorry, I have an alarm. That was my timer ringing. But yeah, speaking of timers, we thought it'd be nice so that everyone sort of knows how much time we have left on each presentation, so we can remind ourselves. Sharing a Koku timer here, so you can join us to yeah, help us, facilitate a smooth call. All right. Next, we have Pradeep. Whenever you're ready.
So are you able to see my slides?
Yeah.
Yes, thank you. Thank you, Amy. So hello, everyone. Good afternoon, good evening, and good morning from wherever you are. So today I'm Pradeep Iranti, a PhD student at the University of Paris. Today I'm going to present my journey in OLS2 master cohort about working towards open platform for Indian bioinformatics community. So the problem we want to solve in this journey was it's creating an alumni. It's creating a community for alumni student interactions at my alma mater. So we noticed that our department did not have a scope for that, where the current students are not able to interact with the alumni and thereby losing knowledge on what's going on in the opportunities available once they graduate. So me along with one of the junior, we set up a pilot project in organizing these interactions and found it to be successful. And then so we wanted to establish and using a community using the OLS2 by knowing the right principles and tools. So that's led us to motivated to apply for OLS2, and the motivations from the program was to learn about openness and the tools required for developing a open project, and then also to get to know like-minded people. But due to COVID, the university got closed, and the university did not encourage for these kind of extracurricular activities for the students, keeping their mental health in mind. So yeah, this happened in the second week of the OLS program, and then we were looking for alternative for a couple of weeks, and then we found out we come up with a situation so challenging, to overcome this situation. So by to understand what are the challenges present in the Indian bioinformatics community, especially the students and the early career researchers for working in open or studying in open. So we created a survey to understand the problems by them, from their side, instead of of creating a platform with my understanding into the picture. So we reached out to the popular personality in Indian academia to share the survey, and then also reached out to the programs where this is bioinformatics has been available. Then we got 70 respondents across different parts of, luckily across different demography, south, north, and east parts of the India. So, and also good amount of students and early career researchers, PhD students, and different areas. So it was a good response we have got. And then we found out 50% of these students or early career researchers did not have a knowledge about open science and how to utilize these benefits in their studies or research. So we thought this as a good opportunity to create awareness in the open science for these students and take benefits from them, and keep them also into the part of the development of the project, in building the community, for at every step, first understand understand the problem, discuss what kind of the solutions available, and then take feedback from them and update, and then release each solution into the public and the community. So unfortunately, due to the lockdown stress levels, the project has to go in pause for 3 weeks. And then so the last stretch of the deliverables and goals of my project were put on hold. So now in the early 2021, we are going to release our website where for the community. And then basically the step remaining is to find the name. We also want to take feedback from the community, what kind of name would be apt for them and how it is easily identifiable. So these kind of suggestions we want to take from them, and then after incorporating these suggestions, we will make it as official release. And also we will also be releasing a handout on open science for students and early career researchers, with an aim that this would serve as a first step for their open science journey, and also to involve like-minded people into the community building. So, and then I want to sum up my OLS2 learnings in 3 categories. One is how I learned how to develop a project using open science principles, and also what kind of things to be kept in mind while designing an inclusive and a welcoming environment community for the to involve more people, and also how to take self-care while developing in the open science world. And then for if you are watching this, if you are interested in developing creating awareness in the Indian bioinformatics community, feel free to get in touch with me. Until January 2021, you can find updates in the GitHub issue, or reach out to me on Twitter, and then we will release the website by January 2021. So, and I want to thank my mentor, David, and the OLS organizers and the cohort, everyone who was part of in journey and to be in future as well. Thank you very much.
Thank you very much, Pradeep. Yes, great work. It doesn't sound like it was on pause. A round of applause for our Pradeep.
Thank you, Amy.
Yeah, loads of folks just really impressed in the Google Doc as well as in the Zoom chat. All the best with the next year.
Thank you very much.
Without further ado, we'll have our next presentation from Georgia.
Fab, thanks. Let me just share my screen. Can everyone see this okay?
I don't see anything yet, I think.
Okay. How about now?
Yep, we see your, yep, you're in presentation mode now.
Thank you. Hi, everyone. My name is Georgia Aikenhead. I'm a research associate at the Alan Turing Institute, and I'm actually here with a collaborator on the project, James Scott, who's going to do some of this presentation. He, in his professional life, is a strategic leader and policymaker, and he's been a wonderful support throughout this project. So I'm really delighted he could join us. I'm here to talk to you about AutSpaces, which is a citizen science platform. This is its working title, it might change, and it's a collaboration between the Alan Turing Institute and Autistica, which is a research charity. So the Alan Turing Institute does AI and data research. And here I have a basic overview of the platform. The idea is that it will investigate sensory processing differences, and people will tell their stories about how this affects their daily life. So this is for autistic people to basically share stories that we can then put into a data set to help adapt environments and make them more inclusive. So James is now going to talk you through how the platform works.
Thanks, Georgia. And it's a very simple process from a user basis. So you sign up, you create a profile, you share your, add in your story. And the interesting bit is around what happens after that. So you have control over your data during this process as part of a fine-grained consent model. So you can choose for your data to be made public, where anybody in the internet can read it, or you can choose for your data to be kept private. And so it will only be seen by researchers. It'll go to a different space on the platform. And the interesting bit is at any stage of the process, you can rescind your permission for your data to be public, at which point it will simply revert to the other parts of the platform and only be viewed by researchers, which is actually quite a novel way of ensuring that people are in control of their data.
Fantastic. And I should point out this cartoon as well was co-designed by James, who made some alterations to make it more inclusive. So actually this project was already in development when I joined OLS2. So I had three main goals as being part of it. One was to grow the community and really connect people up. The next one was to develop the platform or find a means to develop it. And the third one was to share learning. So first off, community. We had a good foundation to begin with, all of these wonderful autistic volunteers who have completely different areas of expertise. And we also had like quite a high rate of motivation and sort of satisfaction with being involved in the project. So this was where we were at. But the most important thing to do was then to map the project out. So this is all of the skills and resources needed. This is useful for onboarding because I can just show this to people and they can like follow their interests and you can see the range of things. And for everyone here who may want to collaborate on the project, I've put a link to this where it sits in the repository in the Google Sheet. So please do explore. Also, this was a category for the hidden ref that was submitted. The hidden ref basically exists to acknowledge people who are usually not sufficiently credited for their work. And that includes people who may have, for example, neurological differences, different experiences, be from underprivileged backgrounds, or just do roles that usually don't fit into the normal ways that we reward things. So this is now a category. Please nominate people that you know who have lived experience, like how they exist in their daily lives. That's actually really vital for science, and it can be a very challenging thing to bring to scientific research as well. I'm going to skip through this very quickly, but basically we did find a developer through this process and onboarded them through all the support that I got from OLS2, who was very excited to be a part, and he's done a lot of work. So that's been brilliant. And the final thing that I wanted to talk about was sharing learning. So myself and James are currently writing a chapter for the Turing Way. Most of the content is there. We need to support it with some of the research that backs that up. But basically, it's in a Google Doc, and you can follow a link and join us in writing it if you want, and it will be in a pull request very soon. This is an autistic collaborator saying awareness is really important to understanding. I mentioned at the beginning that this is a participatory project, which is really important because it means that we're guided by autistic people's needs and desires. And actually, James is one of the people who has been working on this chapter, and he's going to now give his view of what it's been like being involved in the project.
Yes. So, I mean, I think I was attracted to this project because as a non-neurotypical person, I think it's of exceptional value, and it's really, and I find that a lot of research is not really directed, sort of, seems to always be at people rather than sort of involving them as an intrinsic part of the process. So actually being involved in a project where the participation isn't an add-on or a comms piece, it is at the kind of absolutely raison d'etre of the project, I think is really, really good. I've really enjoyed working with a very diverse range of people, people from kind of people in the community, the parents of autistic people, people with technology background, legal background, policy background, strategy background. I mean, having all of those different voices in the room has made it a really interesting and dynamic place to a project to work on. And also, I think, has made the platform fundamentally better as a product, more accessible, and ultimately, I hope, more successful. So that's really excellent. I'd also encourage anybody who is interested to be involved on anything, really.
Yeah, thank you so much, James. James has done multiple different things. He's been involved in different areas, which is fantastic. So I know that we are probably running out of time. So this is just 3 of the main reasons why we would recommend writing a chapter for the Turing Way and why it fit really well with this project. It both spreads something that we believe in as a community that we think should go further, but also allows us to get support from the wider community. And the way that it's written and the content is really aligned with our values. And I really wanted to say thank you so much, Olas. This has been totally wonderful, and particularly my mentor, Inelda, who has been totally fantastic and has just been brilliant to be able to work with you and get your support. So thank you, everyone. Join us, please.
Thank you so much, Georgia and James. Just Malvika says, wholesome approach, round of applause, lots of love from everyone. Yeah, just totally fantastic work to see that you've onboarded contributors, involved participants in such an inclusive way as well. Thank you for sharing what you've learned in the process. I will pass now over to Malvika.
Thanks, Emmy. And yes, I'm just trying to understand everything because it's just so amazing that within such a less amount of time you all have done so much. So I'm gonna give the stage to Beatrice.
Great, thanks. Let me share the screen. Okay. Can you see the slides?
I see it, yeah.
Okay, great. Okay, so my project is called Growing the Galaxy Community. And my mentor has been and still is Dave Clements. So for all of you that don't know what the Galaxy project is, Galaxy is an open source platform for data analysis. And the main value of the Galaxy community is to make the computational research accessible for everyone with a special focus on reproducibility. So Galaxy is a global effort. There are many servers, private and public ones. There are 3 big ones, one in the US where Dave is settled, another one in Europe, this is where I am, another one in Australia. So in September, I joined the University of Freiburg. I knew I took a new position. The goal was to help Dave, which is, as I said, in the US, and he has been doing this job for many years. For such a large community. So for me, joining OLS was also starting a new full-time job. So the project was more or less my complete job. So the first idea was getting to know the community because I wasn't a part of it before. And I needed to know who is doing what are the needs, what do they wish for the community. So I was joining the calls and events. This is what I've been doing this time, also running the Elixir Galaxy community calls, and also updating people with the news and the events and the different websites in coordination with Dave. I also wanted to know here what happened before in the research group that I joined, that's in the University of Freiburg. And so I was writing this activity report for the last 5 years. And so in this particular goal of the project, Dave was crucial because he introduced me to many people. He also gave me some background of what happened in the community before. So I was reviewing my OLS proposal when I applied, and one of the things that I had there is that I wanted to build the community manager skill set. And And again, I've learned a lot here from Dave, because this is the first time that I do this job. So we've been focused on supporting the community, connecting people between themselves, but also with some infrastructure projects, helping them with writing grants and running some online events, and also giving some value to the goals that they achieve. So to coordinate all of that, we have some communication channels. So internally, we chat, I would say, every day. We have shared folders, calendar with events, and we have a Trello board to organize the work too. We also have calls, so every week we catch up, both of us, but also we have a weekly outreach meeting with the Galaxy PIs. And this, me joining here, has motivated a change. So now we are doing it globally, and we've created this outreach team. And in these 4 months, we've been working also with Christina Hall, which is in the server in Australia. And we've been also running calls every month with her, the 3 of us. So this is more or less for the internal communication, but we also needed to communicate with the community. And for that, we have several Twitter accounts and different mailing lists. Okay, so and also the main point here was to do outreach. And we've written several articles, and there's also some preprints out there. Dave was doing before the monthly newsletter. So every end of the month or beginning of the month, there is a Galactic news that goes out. Yeah, so I've been helping him in the last months to add their events, open positions, publications, and all that's going on in the community. And yeah, so OLS is just the beginning, because I believe that we've created a solid team. We are running, for instance, today the Paper Cuts Day, in which we welcome new contributors that can learn from the experienced core developers that we have. And we've been doing that for 3 months already. So the whole day we have calls for onboarding people. We are also involved in organizing different webinars, and in July we will have the GCC 2021. So this is the Galaxy Community Conference that will be in July. So we are also involved in the organization of that. And I think this is my last slide. Thanks, Dave, for all the help, and thanks for OLS for the opportunity to participate and for learning so much.
Thanks, Bria. Can we all have a virtual round of applause for her, please? That just doesn't sound like something that could be done in 4 months. So I am very impressed. So I'm gonna move on to our next speaker, Kariga Pauline.
Hello. Hope you can all hear me.
Yeah, perfectly.
I'm going to share my screen now. Right, so my project is called the Hub Portal. Basically, the Hub Portal is an extension of the Bioinformatics Hub of Kenya. The Bioinformatics Hub of Kenya was formed last year. It was a part of the OLS1 cohort, and this year we saw the need for it to grow, so decided to join the OLS2 cohort to be able to learn and to be able to expand the Bioinformatics Hub of Kenya. So the Hub Portal is a resource platform found within the Bioinformatics Hub of Kenya. BHKI is it's a community of bioinformaticians that brings together bioinformatics students and also mentors or experts in the field. So the Hub Portal is the resource platform part of BHKI, where basically guys can find all the tools that we talk about in our trainings, in all our meetups. That is where everything can be found. Yeah, it's a resource platform. It's a website. So the objectives that we had as the Hub Portal, or BHKI extending to the hub portal for the community, just to be able to provide a resource, not just for the people who are on Twitter, but also just have it have a one-stop shop where everything can be found, which is what we wanted to do for the community. And personally, as a member of the OLS2 cohort, my personal objectives, one of it was to be able to learn and understand GitHub, which I was able to do, and I'm very grateful for that. Yeah, so some of the challenges that I faced, especially within the first phase of the project, was being able to see the bigger picture of the project. Despite the fact that we are a community, or the BHKI members, we are 5 of us, being able to see the broader picture of it was a bit of an issue, and to be able to break it down, to be able to develop it was a bit of an issue. But thanks to Lorena and my mentors, I was able to overcome that challenge and be able to break it down well and to be able to develop it step by step. Communication also was a bit of an issue, but I was able to overcome that as well. So the achievements that I've been able to make for the hub portal is I gathered the resources that I needed to build up the whole platform. I have links in the portal, links for learning. We have a YouTube playlist on there as well for bioinformatics. We have coding tutorials. We have links for downloading books. We have practice coding platforms. We have research institutions in Kenya where our students can be able to apply once they've been able to learn all these things. And we also have a section where it's an opportunities alert, PhD programs, master's programs. We'll be able to announce all that in that area. So the what next for the hub portal. So this is like an open call. If you have any resources that we can be able to use or put in the hub portal, the contribute.md is very detailed. You can just read it and add on to the resources that are on there. Anything that you have, will be very much grateful. And also we're looking for ways to make it more collaborative for the students that we have, or for the members of the community that we already have, and to be able to invite more. So I'd like to acknowledge my mentors. In the beginning, I had Lorena and Sarah Gibson. They've been very much helpful. Some of the BHKI members, Festus, Nasimi, and Michael. I think Festus is in the call today, and the Open Life Science community for the opportunity to be able to be part of this. Thank you very much. Yes, thank you.
Thank you so much, Pauline.
And follow us on Twitter and GitHub. Thank you.
Amazing. Can we all have a virtual round of applause for Pauline, please? Amazing. Next group of people are Paul and Neha.
Hey everyone, just gonna share my screen.
Everyone.
Alrighty, do you all see it? Awesome, okay.
So, hi everyone, my name is Paul Oecho, and together with my project partner Neha, we're gonna talk about how we embedded accessibility in the Turingway open source community guidance. Next slide. So for those of us who don't know, the Turingway is an open source book project that seeks to teach people reproducible, ethical, and collaborative data science. And by virtue of the fact that it's an open source project, it has a very wide and diverse community behind it with over 200 contributors at the moment. And while that's a good thing, the variety of contributors has also introduced some inconsistencies in the way that the book is written, in how it's formatted, and generally how it looks. Um, and this inconsistency also led to some accessibility issues. So on the one hand, for new contributors, the book became slightly more challenging to contribute to because it was difficult to kind of follow the book style guides when existing content in the book didn't follow them. And for readers, there was some information loss when they tried to read the book on accessibility devices such as screen readers or even just mobile phones. So to address these accessibility challenges, we had three project goals. The first of which was to look back through the existing content of the Turnway and figure out how to make it retrospectively consistent. Secondly, we wanted to lay the groundwork so that keeping that consistency was easy to do and also make it easier for new contributors to keep contributing content to the Turnway. And then finally, we wanted to reduce or rather minimize the overall information loss that readers could possibly experience while reading through the book. And all of this was not without challenges. And my project partner, Neha, will tell you what our challenges were.
Okay, thanks, Paul. So with respect to the challenges that we faced during our project, the first one was just getting up to speed with OLS2 and the Turing Way. We only started OLS2 in week 4 of the 15-week program, and we had some great onboarding by Malvika and our mentor, Sam. But we were also new to the Turing Way project and community. So in general, it took us a bit of a while to just get oriented, figure out all the homework and what is the timeline and what we were going to be doing. The second challenge was figuring out how to collaborate efficiently between the two of us. Paul was working for the Turing Way full-time through Google Season of Docs, while I had just started a new full-time job, and I set aside about 2 hours per week initially for the project. So we really had to figure out how to sort of keep up with one another and kind of stay in touch, communicate, and follow up and help each other out. The last one was just learning how to work effectively with GitHub. Both of us were familiar with GitHub, but we were working on such a big community-driven project that we really had to improve our pull requests and our issues so they were just accessible and readable for the contributors so that they can really like work with it, pick it up and work with it. So from these challenges, eventually we did finish everything and we were super delighted. And there were some lessons that we picked up and we would love to share. The first one was we should probably maybe anticipate some trial and error in open source projects. You might have a really lovely roadmap, but maybe things don't go like linearly, but you're kind of going a bit in circles, back and forth, left and right. And that's okay. The second was that you just have to be really patient and flexible, both with yourself and your collaborators. Eventually things will click, but it might take a while. And as you figure that out, just be kind to one another and try to have fun with it. It's going to be frustrating, but just kind of go along with it. For us, it was a Turing Way book dash, which really just sort of got us on track and just really made the whole thing happen. We, of course, learned a lot about what goes into building a good open source project and a community. We learned a ton through OLS2, and we also had the chance to just see it applied in real life in the Turing Way. For example, a nice README, contributing guidelines, code of conduct, all this stuff. And the last one, and this is both a lesson and a take-home message, it's like, when you do an open source project, it's not all about doing the work yourself as an initiator, but really also about empowering and enabling other people to become an equal contributor just like you. And with that, we would both like to acknowledge all these lovely people. These are our mentors and also the Turing Way, a community manager and project lead, and everyone who was on the book dash and in OLS2. Hearts for all of you. Thank you. And that was it. Thanks, everyone.
Thanks, everyone.
Thank you so much. I might be biased because I think they are amazing. It was great working with both of you. But please, let's give them a round of applause. Again, the virtual And before I get more emotional, I'll give it to you.
Okay, we've had some fantastic presentations so far. We have, I think, about 4 left. And next up, Sangram, are you here and are you ready?
Yep. So let me share my screen. Let me know if you can see the screen. Okay, cool. Okay, hi guys. So my project is about like it's a practical guide to reproducibility in special in bioinformatics. So it's a handbook and like online handbook and towards a community. So yeah, what's the origin basically? Like so in early 2020, like I was reading a lot of publications. So even I was trying to reproduce few of them, but it was very challenging when I dig down a little bit. So even there was a news like in Nature News in 2016. It was in 2016, but it holds true to this date as well. So let me figure out, like, let me tell you, like, what reproducibility actually is, like, what we are focusing on over here. So anything to do with like obtaining the same results from the from conduct of an independent study whose procedures are as closely as matchable to the original experiment, right? So yeah, so this was really difficult. So especially in our field of bioinformatics. Uh, so yeah, even if you see like the one of last year publication, so it is straightly mentioned that reproducibility is a crisis in the age of digital medicine, especially. And like although the scenario is changing, there are a lot of tools and methods are available to do the reproducibility basically. But if you see in practice, there are a lot of things are missing. Like some people like find it difficult to use, or sometimes people just don't use it for their accessibility. So for that, we wanted to create a practical approach to the reproducibility, especially in our bioinformatics web. So if some of you from here don't know about bioinformatics, so bioinformatics is a field like where we use a lot of data science tools to answer biological questions, right? Like especially in this time of COVID, we're using this SARS-CoV genome to get to the lot of informations about this virus, right? So that's what we do. And literally, if you can't reproduce a experiment, then it's a lot of like mis experiments basically, right? So yeah, so for this we're trying to create a handbook. And uh, yeah, so that's our journey basically starts in OLS. So when I entered the OLS, like I just had idea and then few very initial things put on place. But after joining the OLS, like finally it made their way into a real online book, which is like already deployed. But this is gonna change a lot with their like visualization and everything. So yeah, also like to get the community to contribute, because like there are lot of chapters we're gonna write, and I can't do it alone. So we wanted to have the open source community for this. And we made the GitHub repo welcoming to the contributors based on the calls we had for all the like how we can make a contribution.md, like different approaches, open approaches basically. And also during the journey, especially like thanks to my mentor Hans, who really put the picture. Like I had a very different, like which might not be the realistic one, but he really planned it very well. Like I was thinking to write all the chapters from beginning to end, but his idea like to cross-referencing and adopting which already existing materials, which is a really good thing, good suggestion that we took. And even thanks to Malvika, like I joined the Steering Way Book Dash, which happened last month. And over there, I got a chance to create a case study for bioinformatics into the Turing Way book chapter. And also like discuss lots of amazing stuff to basically interchange the intercombine the materials basically. So yeah, like what I'm gonna have going forward, like I'm gonna write the practical approaches on my book, and for theoretical and other parts, I'm gonna just redirect to the training web book, because that's very well written by the amazing community of people, right? So that's the journey so far, and going forward, like I want to redesign the book basically, which can be more approaching, like bit, if you go from different resources, it should capture the same thematic style, which I'll be doing in coming weeks, even extending the chapter. For now, we have very few materials, but going forward, like I'm planning to welcome my friends to participate in it. So we're gonna extend the chapters as well. And even going forward, the one thing we really want to put in place, like hosting talks around the book, basically, so that it can really touch the researchers who want to use it in their day-to-day practice. So yeah, that's it for it. And I want to thank like Hans basically for being supportive and mentoring throughout this journey, and the OLS team, especially Yuri and Malvika, and the Tuning Web project, which hosted me for the book dash basically. So thank you all.
Amazing. Thank you so much, Sangram. And thank you also everyone who's putting comments in the Google Doc and questions. So keep those coming in. And can we have a huge round of applause for Sangram?
Thank you, guys.
Awesome. Okay, now next up we have an OLS grad repeat with a new set of people. Awesome team, are you ready?
Yes, we are. So yeah, like thanks you very much for having us. And like you mentioned, this project was started at OLS 1, and this new cohort brought in new people, myself included, and we've expanded the team, and we're moving forwards. And so just to give like a brief overview of what it is, essentially we're trying to create this onboarding to open science, but in French. And as you can see in this cartoon, you know that it's hard enough trying to convince scientists to do open science, but there are communities who are francophone rather than anglophone, and helping these people get into open science is a challenge. And one of the reasons we're doing this, if you go to the next slide, Sam, you can see that there is large areas of the world which speak French, and for us here in Montreal, it's the case. And for those of you who might be familiar with the dynamics of the Welsh or maybe the Catalans, the language is a strong part of an identity, and that's very true here in Quebec, where it's a hot button topic. So at the University of Montreal, we're really trying to drive inclusivity in open science by offering open science in French. And slide, Sam. And one of the key ideas is how do we do this? Well, the first thing we really need to do is to evangelize open science to Francophone communities. Once we've got people with the bit in their mouth, it's to onboard to what it is. As many of you know, it's very demanding and difficult to really know how to start doing science in the open, and how can you help people to start tasting from that broad menu that's available. And so our hope is to generate content as well as in-person training, bringing people into science. And finally, the big goal is also to create the community, to create visibility of open science, and to really drive that change that we all want. One of our great experiences during OLS2 was our mentor, Renato, who I think is here. He was great. Really enjoyed having him on. He was always adapting to what we needed and was very accessible. Was a great listener, which was fantastic, and entertained a lot of the tangents I like to go on, talking about philosophy of science. But most of all, he really offered a bunch of really useful tools to help us tackle these big obstacles. And you know, those big ones were organizing, mobilizing the people who are interested. So people who are interested, how can they help? One of the things that really helped was also prioritizing and delivering from all the things that we wanted to do. And then finally, what we actually achieved during the session, which was really the great success, we will give you a mini tour in a moment with Sam G, another member of the team. But we managed to get our MVP of our website up and going. We opened our own matrix chat for Open Science Montreal in Canada. We met our mentor, who was awesome. Sam and Adrian presented Open Science in Montreal to the nurses of Canada. And we've really broadened our team. And we also met Kendra at McGill, who presented yesterday, who was doing the Open Science Office Hours. One of the other great successes was what Sam is about to show you now is this MOOC that he contributed to heavily for EduLib. So I'll hand over to Sam.
Yeah, so EduLib is a French MOOC platform like edX or Coursera. And they, a professor there asked us to produce, it was supposed to be an hour of open science content, but then it ended up of being 2 hours of video content on all things open, from reproducibility to Git and GitHub, and some hands-on exercises. And all the awesome community really contributed to the content, which was really amazing to see the small community we have, but then put effort and produce that kind of work was really rewarding. This MOOC is going to open in February 2020. So the EduLib was really happy about this, and they even asked me this morning if the awesome community could produce a full-length open science MOOC, which is really encouraging. So we were really happy about this. And I guess we can move on to maybe showcase a little bit of the website. So basically, we've redone this all template that we had to produce bilingual content. So everything is working now, which I'm really happy, and we can see that we've grown our community a little bit, and it's still, some emails are still coming in, so we're happy about that. So I don't know, Sam, if you want to give the final thank you to everyone.
Yeah, I think it's just that. Thank you very much for what you're doing at OLS. I think it really helped to concretize trying to make things happen, and it really helped us get things done, as Thingy Alan would say. So thank you very much.
Amazing. Thank you so much, everyone. These projects are just also inspiring, and maybe we can say OLS in French one day. That would be like a real winning moment. Huge round of applause, everyone, for awesome. They are indeed awesome. So, Taina, are you ready?
Yeah, I'm ready. So let me share my screen. That's it. So hi everyone. I am Taina Rocha from Botanical Garden of Rio de Janeiro, and I present my final project called Global Land Use Land Cover Data under Historical, Current, and Future Climatic Conditions for Ecologists. So we have two main goals with this project. One, it makes more accessible the land use harmonization data. Once these data are in XCDF format, that's not so manageable for most ecology analysis, because NetCDF is something like that, like a cube of too many layers. And for most ecology analysis, we need one single layer. And also we provide a new data from the raw data, something like that. I will show more. So the land use harmonization database provides classes or types of land, something like forest land, pasture, rangeland, urban land, crops, all these. So I worked on it because mapping the land use and land cover changes through time is important to predict the threats or propose effective conservation policies and many other kinds of ecology studies. So, however, most databases have a time series limitation. And contrary to these databases, land use harmonization covers historical, current, and future land use projections. So it's a really complete database. And to do this, I built our scripting following these steps. One, to extract each single layer from etcdf, and after I save each one in a TIFF format, that is more common format. And also I perform an analysis to combine all classes to generating this data, a land use and land cover data that mixes all these classes. And finally, now I need to validate this new data, and this step is still in progress. I'm working on it. So all materials of my project are available in this website in GitHub. So feel free to visit this repo. As a result, we have several continuous layers for each class and for many years. As an example, this is a raw data in a TIFF format. For forests, for example, range from 0 to 1. So this is a raw data, and this is a new data. We have several categorical layers for many years. For example, 2010 year or 2100 year, mixing all classes. In the next step, I need to optimize the R script, and also it's a challenge. And also I need to improve the README. And finally, I need to put the layers in EcoClimate database, another free database, a Brazilian initiative of databases of climate. So I need to say thanks to all OLS team for the knowledge shared here. It's a really good materials. And a special thanks to my mentors, Bruno and Ivan, for all support. It's really important. And that's it. Thank you very much.
Amazing. I really loved watching those maps change. So huge round of applause, everyone, for Taina.
How can I stop the share?
Is there a red button? Oh, you've got it.
Oh my gosh.
Awesome. Okay. Fantastic. Sebastian, you are on the ball. Go for it.
Thanks.
Hey, now I think you can hear me. Hey, I would like to welcome you also from my side. My name is Sebastian, and within this talk, I'm going to highlight my journey throughout the course and also introduce you to my project, the Open Workstation, and how I want to democratize access to lab automation. So first, let's start with the motivation for my project. If you go back to the 50s and 60s, if you look to the labs, you could still find a lot of manual workflows such as pipetting. What are the limitations? As you can guess, it's not only time consuming, it's also rather low throughput. And the consequences, as you can also imagine, it's not efficient because you have to do a lot of tasks manually. And if you want to screen various different parameters, it's only limited scalability. And as the human factor is involved, we also have issues with reproducibility. However, however, nowadays we have various of different lab automation devices commercially available, which are able to increase the throughput as well as the efficiency. But actually, let's wait. If you take a look into academic labs, you cannot find those devices, and most of the stuff is actually still done in a manual-based manner. So why? Actually, it comes back to these three reasons. So first of all, automation is expensive. And as academia is working on a grand budget, the price becomes prohibitive. And secondly, and this is also connected to the third point, as we as scientists, we want to try new ideas. We want to customize our software as well as our hardware, and we want to have a flexible setup in order to adapt to new and emerging technologies. And therefore, I built my own research platform. I built my own automation devices, which you can see on the right side. So there are different modules, different platforms. And this is actually the point where I heard about Open Life Science course and where I signed up. So the motivation or the aim for my journey at the course was to make the entire tool set which I have developed, make it accessible to all other scientists, to build an open source project based on the tools and methodologies I've developed, and also learn how to do outreach and to build a community about this platform. And overall, my vision is to democratize access to provide lab automation to everyone who wants to have lab automation. And therefore, I came back to the platforms I developed and revised my concept and built everything on those 5 principles, which are a do-it-yourself approach, a modular setup, open source, low cost, and systems engineering, which is actually a design methodology in order to guide the engineer throughout the development process. And what I want to highlight with this slide is that I've developed a kind of a tool set and a guiding path in order to help you if you want to develop your own automation platforms. So for example, there's dedicated information for the different modules. If you want to use a Raspberry Pi for the computational module, and there is information how you can use the Raspberry Pi, how you can connect it, and also how you choose the aluminum profiles, for example. Throughout the last 4 or 8 weeks, I was able to put together the open source project. I was able to release it on GitHub, which was actually quite exciting for me. So all the hardware and software files for the different modules are now available. There's also an installation guide and a documentation, and there are also the experimental protocols available. So it's kind of a platform which not only provides access to hardware and the software files, it provides also access to the experimental protocols I was using. And this is an important point in order to make science more reproducible, as we have learned throughout the last presentations. So what are the challenges which I identified or which I was facing? Although it sounds simple, and it took me actually quite some time to realize it, and the end users and the biologists, the chemists, are often not the developers. So in my case, the developers are the mechanical or the electrical engineers who build their platform, who assemble the different parts, but in the end, the users are different. So this comes also back to the second point. The skills that you need in order to build this platform is very interdisciplinary. And I want to address those outstanding points or those challenges by providing additional materials on how to assemble the platform and how to put together the materials by, for example, brief videos on the guidance. And finally, for myself, it was also challenging to find the time to find the time to work on different projects. I had several ideas, but not only due to COVID, but also due to my job situation, I was not able to invest more time in the project. And this is what I want to change in the future by applying for separate funding in order to realize this project. And with this, I would like to thank you, thank the entire OLS organizing team, thank my mentor Meg, and also thanks to the OLS community. And if you would like to join, or if you would like to talk or get any input, feel free to send me an email or join me on Twitter. Thank you very much.
So the science wasn't doing what I needed, so I took it into my own hands. Can we have a round of applause for that? That was just like, yeah, amazing. Folks, we've come to the end and we're only 8 minutes over. I honestly consider that a win. This has been really fantastic. I've loved every single one of these presentations. And everyone's been really lively in the Google Doc with the comments and the questions. So please do make sure, go and check your projects and see if there's any comments that you maybe want to answer. This is the very single last call of OLS2. There are no more graduations coming. We love you to pieces and we've had such a great time and we hope you have too. Emmy, Malvika, anything you want to throw out there?
No, the reason you're the last person hosting this is because we knew you can capture all our emotion perfectly.
Okay, one last round of applause for every single person, every mentor, every co-host, every project leader. And yeah, oh no, I need to go away now. I'm definitely getting emotional here. It's been so delightful, and everyone have a great time, and maybe we'll see you as OLS3 in some other capacity, and we'll definitely see you in Open Science and see you around.
Should we take a group picture for people who would like to?
I'm so glad you asked that before I dismissed everyone. Okay, folks, if you don't want to have a group picture, please turn your camera off. If you want to have an awesome weird face, now is the time. And hold that grin. I've taken one screenshot. And I will paste it at the end of the doc. And there's 2 pages, so don't go away yet. And I'm going to move to page 2. And put on the awkward grin again, or the awesome face. Okay, I got you all.
There is Meg with Santa hat.
Do have the Santa hat. That has been captured twice. Awesome. You are the best people. I love you. Goodbye. Have a great day, evening, morning, all.